All right, so we are on the last lecture of the first unit already. First unit is lecture 1 to 5, and number 5 is actually quite complicated. All right. There are a lot of information for lecture 5. Uh, I will try to break it down into smaller chunks for you to remember. And when it comes to the tests, I will try to narrow it down even further for you so that you can focus on specific aspect of lecture 5. But for me to teach it to you more effectively, we are not going to be using the PowerPoint slides, okay? The PowerPoint slides are still important for you to read through later on as like supporting materials, but I think we're just going to be taking a huge note. We probably won't be able to finish it today, but that's fine. We'll whatever we don't finish, we'll finish next week. Next Monday is the lab though. I'll talk more about the lab later. Specifically, I put you guys into the groups that you wanted to be in. Some of you didn't have a group. So I just put you guys into 7 and 8, and most of these people are not here at the moment. So maybe when they get here, uh, I'll put this up again. But the lab is on Monday. There are lab sheets right there, the green one and the pink one. I'll tell you more about it when more people get here. And you do need to be on time for the lab, okay? There will be a 10% penalty for every 10 minutes that you're late, okay? Because it's not fair for your classmate to start the lab and then you just join in, middle and then, get the same grade as them, right? But it's not going to be a problem for everyone here right now because you guys are all on time, okay? I'll repeat this message when more people gets here. Anyway, so we're going to be doing lecture 5 today, and you really need some color with the notes. It's not going to be as crazy as those, cell organelle pictures that I drew, okay? But you get one of these pens, okay? The Centennial ones. Okay? They have 4 colors, and nice, happy colors that make your notes look amazing. The lecture is called Metabolism. Okay, as I said, this is the hardest lesson probably for the entire unit. So please ask questions if you are stuck. So let's start with the reaction, cellular respiration. Remember, cellular respiration is the key reaction in a cell that produces energy. And the usable form of energy in a cell is ATP. Right? So the overall reaction goes like this. And please, please, please memorize the reaction for the test. Right? So we start with glucose, which is what we will obtain from eating carbs. And we are going to be reacting it with oxygen, which we breathe, to produce carbon dioxide and water, as well as energy. And so this is the word equation. By now, you should have learned about word equation and chemical equation in your chemistry class, right? And so that's no? Okay, that's all right. A word equation is a reaction that's written in words, okay? And then a chemical equation is an equation that's written in chemical formulas. So for example, glucose, the formula would be C6H12O6. And what's the formula for oxygen? Does anybody know? O2, exactly. O2. And it's going to produce carbon dioxide, which is CO2 plus water. Did you guys learn about balancing chemical equation yet? No? Okay. So when we do a reaction, the number of atoms in the reactant side must equal to the number of atoms on the product side. Right? It's like if you buy a box of Lego, you can use the Lego pieces to build a car, and then you can take it apart and use the same Lego pieces to make a plane or something. Even though the two things look different, but the number of each type of blocks remains unchanged, right? So over here, if you take a look, we have 6 carbon here in the reactant, but we only have 1 carbon over here, right? So it's not balanced. You're missing some atoms, right? So exactly how you balance out a reaction, you will learn in chemistry. I will balance this one for you. Okay, so it's 6 for the oxygen, 6 carbon dioxide, as well as 6 water. And so briefly, we can check. For example, now we have 6 carbon here and 6 carbon. How many hydrogen do we have in the water? 12. 6 times 2 is 12. And over here, we have 12, right? And finally, we have 6 oxygen plus 12 oxygen. That's 18. And then we have 12 plus 6, which is 18. So now we have a balanced chemical equation where the number of atoms are the same on both sides of the equation. Okay? Once again, I need you to memorize this equation in the balanced form for your tests. So, more importantly, it's going to produce energy. And as I told you, energy is captured and stored in the ATP molecule. Under ideal conditions, the metabolism, the breakdown of one glucose molecule is going to be able to give you up to 36 ATP. Okay? Up to 36 ATP. Okay, so a couple of notes here for cellular respiration. This is a redox reaction. This is a redox reaction. Where electrons are transferred from one reactant to another. All right, sorry, I'm just going to give myself more space here. So let's break the word down. Redox. Oops, redox. Okay, so re here, that stands for reduction reaction. So reduction means gaining electron. It gains electron. Because we're going to be writing the word electrons like 10,000 times today, I'm just going to do that for electron, okay? You can do that too. Okay, so that's what the red, okay, represent. Reduction reaction, which means gains electron. On the other hand, the ox, that means oxidation. Oxidation is loses electron. Okay. Some people like to remember it like this. Oil rig. Okay. Oxidation is lost. Reduction is gain. Okay? You guys know oil rig, right? Like those big things in the middle of the ocean that, gets oil. So whenever something is reduced, it gains electron. Whenever something is oxidized, it's losing electron. So not every reaction is going to have this transaction, but for those that do, we call them redox reaction. So let's go back to our overall cellular respiration equation here. What happens is we essentially will be oxidizing glucose. Okay, so glucose will be oxidized into carbon dioxide. So this is loss of electron, oxidation. Okay. We will be removing high energy electrons from the glucose. And ultimately, those electrons are going to be transferred onto oxygen. So because oxygen will be picking up those electrons, it's going to be a reduction reaction, gain of electron. And in the process of harnessing the electrons from the glucose and transferring it onto oxygen, you release a lot of energy. And we can use those energy to produce up to 36 ATPs. Okay. Any questions so far? All right, I'll pause for a moment for you to catch up.
To get the
Electron?
electron from water.
From glucose. Okay. So we're going to take the electron from the glucose and then you're going to transfer it onto oxygen. Okay. So when glucose loses the electron, it's oxidized. But when oxygen picks it up, then oxygen is reduced. Okay. So once the glucose have lost the electrons, then it becomes carbon dioxide. Okay. And once the oxygen picks up the electrons, then it becomes water. Okay. All right. So that's that so far. All right. Yeah. Once again, hint, hint, nudge, nudge, wink, wink. You gotta know the formula. You gotta know what's oxidized. You gotta know what's reduced. Okay. For the test. Okay. When's your quiz, by the way? Friday. Yes. Okay. Don't miss that. Okay. 4 stages of cellular respiration. It turns out, even though when we write the overall equation, it's just one equation, glucose, oxygen becomes carbon dioxide and water, but it actually does not happen in a single step, okay? It's broken down into 4 different stages, and within each stage, there are multiple steps as well, okay? So think of it like this. Your car runs on gasoline, and I told you gasoline is octane, right? In fact, when you fill it up, it says on the button it's octane, right? And when your car burns the octane, it's actually the same reaction, but instead of glucose, you would have octane plus oxygen produce carbon dioxide and water. Okay, so how do you use the fuel to travel a long distance? Well, you do controlled explosion a little bit at a time, right? Okay, your engine injects a little bit of fuel and then you burn it and the explosion drives your car. So that gets you really, really far. Whereas if you lit up the entire tank of gasoline, you get a big explosion, you release the same amount of energy, but in a short period of time, and that's not going to get your car really far, right? So in the same way, we don't want to burn glucose in a single step. That's just going to release a huge amount of energy in a very short period of time. It's going to burn our cells, and then, it's not going to be meaningful extraction of the energy. Okay. So rather, we want to do it in little steps, a little bit at a time, do it as stages so we can harness the energy a little bit at a time in a meaningful way. Do you understand that? Okay. So 4 stages. I'm just going to write the names out first, and obviously I'm going to explain what each one does. So, don't get too hung up on the names yet. They are just names at this point. Okay, so stage 1 is called glycolysis. Stage 2 is called the Krebs cycle. Krebs is a person, okay? Also, sometimes known as citric acid cycle. Oh, sorry, I lied. I'm not fully awake yet. I'm still recovering from the weekend. Number 2 is a pyruvate. Sorry. Hear all the frustrating erasing. Pyruvate oxidation. Then we have the Krebs cycle. And finally, we have the electron transport chain, which is ETC. Okay. Now, which organelle does cellular respiration happens in? Part of it anyways. Do you remember? Not what organ, what organelle. Yeah, like inside the cell, we have like, the Golgi body and everything, right? Which organelle plays an important role in cellular respiration? That's something you have to know for the quiz, guys. Like oval shape, it has 2 layers. Usually it's orange, even though it's not really orange in real life. Starts with the M. Yeah, that's the one. Okay. No hints for the quiz though. Okay. So make sure like I'm not gonna be there, breathing down your neck and say, all these hints. So make sure you do spend some time to prepare for the quiz. Okay. So here is the cell. Okay. And this is the cytoplasm. Cytoplasm. And I'm gonna draw a mitochondria. It's not to scale. Okay. But we have to draw it nevertheless. So the first stage, which is glycolysis, it happens in the cytoplasm. Stage 1 happens in the cytoplasm. Stage 2 and 3 happens on the inside of the mitochondria. Does anybody remember the name of this part of the mitochondria? like a famous movie with Keanu Reeves. You guys know where that is? What is it, huh? The Matrix, that's right. Okay. So step 2 and 3 happens in the Matrix. And step number 4 happens right here in the foldings. Okay. So the foldings, they are called cristae. We drew this mitochondria in lecture 3. So each stage has a specific location in which it happens in. All right. So let's give you an overview of what exactly is going to be happening. Okay, and then we're going to go into a little bit more details. Basically, in the first 3 steps here, in the first 3 step here, okay, high energy electrons are removed from glucose. Okay. Remember, the overall reaction, glucose is the one that is going to be oxidized. When you oxidize something, you remove the electrons. So we will continue to remove electrons during the first 3 stages. We will collect these electrons, which again, it's high in energy, and we'll collect all of them, and then you will transfer them to the last step. Okay. Electrons will be transported to the ETC, to the electron transport chain, by electron carriers. There are any questions so far, by the way? Yeah, it's okay. All right. There are 2 energy carriers that I need you to know, because they will be important for understanding the rest of the lesson. I'll write down for you. There are 2 types of electron carriers. I'll write a name for you. NAD plus and FAD. Okay, never mind what they stand for. I mean, you can Google them. It's a really long name. Doesn't matter. You can just call them NAD plus and FAD. Now, I will write 2 equations for you first. and then I'll explain after. So NAD plus, I am simplifying things here. So, I don't suggest you Googling these things. If you don't understand, just email me, okay? Because if you Google it and you read it, it's going to be like information overload. This is already information overload, right? For many of you. So if you look it up, you're going to see even more confusing details, okay? So, if we simplify it, basically, if you take NAD plus and then you add electrons to it, if they pick up electrons, then they are going to become NADH. Okay. Again, never mind where the H came from. It's just floating around in the cell. Right? So when the electron is picked up by NAD plus, it becomes NADH. I'll give you an analogy in a second. It will help you understand better. That's the wrong color. If you have FAD and it picks up electrons, and it's going to become FADH2. Okay. So what kind of reactions are these? Is it oxidation or is it reduction? One for oxidation, two for reduction. Picks up an electron and becomes something else. Oxidation or reduction? Show me. Good. Good. Yes. Anyone else? Oxidation or reduction? Good. Good. This is reduction because remember oil rig, right? Rig reduction is gain. If you gain electron, you are reduced. Okay? So here, because the NAD plus and the FAD, they are picking up the electron, these are reduction reaction. Okay, so this is reduction. Now the reaction can happen in the other way. Okay, the NADH and the and the FADH2, they can turn back into NAD plus. They can turn back into FAD and let the electron get off. And when that happens, this is oxidation. This is oxidation. Okay, you can watch this part and then you can decide if you want to copy it or not. Okay, maybe let me explain first. So you look at this, what the heck is he doing, right? Okay. So 1, 2, 3, stage 1, stage 2, and stage 3, okay? They are like 3 different hotels, okay? And then we have tourists who wants to get to the airport, okay? The airport represents the stage 4, right? So in this analogy, the passengers, the tourists, they are the electrons, okay? So, we have people here, and we have people here, and then we have people here. So in real life, when people want to get to the airport and they don't have a car, what do they do, huh? That's not a difficult question. You either call a taxi or you call Uber or something, right? Okay. So that's what the NAD plus and the FAD are. They are basically Ubers. Okay. They are basically transportation to get electrons from one place to another. Okay. So we have, and don't copy this yet. Okay. I'll show you what I mean. So we have a NAD plus. Okay. Basically, it's like a car. Right? And then the electrons will hop on. When they used to have the taxis, it's not so much anymore, but like the traditional taxi, like if you see a taxi coming from afar, how do you know if it has people in it or not? Like, let's say you cannot see the inside, right? Like, how do you, yeah. Yeah, the light, right? Like it turns on, it says occupy, right? When there are people inside. Do you guys know what I'm talking about? If not, please Google what a taxi looks like. And so this is what happens. The NAD plus is like the empty taxi. But as soon as the people gets on, right, it lights up, right? It changes the state, right? So it went from NAD plus to NADH, right? Basically, the NADH and the FADH2, they are the occupied version of the molecule. If you see them within H, if you see the light turn on, you know they are occupied. Okay? So they will take the electrons to their destination, which is the ETC. Right? So once they hop on, right, the passenger hops on, and again, don't draw this yet. I don't know how I'm going to finish this note, but they're going to go all the way there. Okay. And then the people get off. Right? People get off, right, and destroy the car the way. And now this is empty. So what's going to happen to this car? All right. What can it do now? Well, since it's empty, it's going to able to go back to one of these places, right? Pick up more people, pick up more electrons, and then come back. So that's what the energy carrier does. It just goes back and forth, shuttling electrons all day long. Are we together? Okay. Stages 1, 2, 3 releases high energy electron. The energy carrier, the electron carrier, they capture them and they bring it to the ETC. Okay. So let me try to draw this out for you in a way that makes sense. So the NAD plus will pick up the electron, and then it becomes the NADH, which then takes it to the ETC. But at the ETC is the electrons are going to get off, and then it's empty again, so it can go back to pick up more. And now the only limitation in this diagram is, just because you came from the first hotel, just because you came from the glycolysis, you don't necessarily have to go back there, right? Like you can go to other places to pick up electrons as well. Okay? So copy it down if you want, or if you already understand it, then that's okay. You don't really have to copy it. Okay, so this is FAD, and then it's going to become FADH2. Now FAD can only go to hotel number 3, okay? Because of things that I haven't told you yet. And so this one's going to go over here. It's going to drop off the passenger is going to drop off the electron, and then it's going to go back for more. Okay, so there is this, continuous shuttling between the first 3 stages and the last stage. All right. That's you. So remember, these electron carriers, after they capture the electrons, those electrons are actually packed with energy, right? So sometimes energy carrier, electron carriers are used interchangeably. But the key thing for you to know here is that each NADH is capable of making up to 3 ATP. Under ideal condition, you will be able to make 3 ATP. And for FADH2, you can make 2 ATP. So here's a concept check question. What is the maximum number of ATPs that can be produced from 5 NADH and 3 FADH2? Okay, simple math question. Feel free to bring a calculator to the test if you need to. But what do you think the answer is? Anybody? What is it? 21. That's right. So it's quite simple. 5 NADH multiplied by 3, because each NADH can make 3, right? That's going to be 15. And 3 FADH2 multiplied by 2, it's going to be 6. 15 plus 6 is 21. That's it. Okay. So there will be questions like these on the test. Okay, so that's the overview. Any of you have any question at all before we get into even more confusing stuff? And again, I'm just a messenger, okay? Like I didn't decide you have to learn all these things. Okay, let's carry on. Of course, I'll post a note. So, if you missed anything, it will be available to you later. New section. Oops. In cellular respiration, which is again glucose plus 6 oxygen, producing 6 carbon dioxide and 6 water, in that reaction, the ultimate purpose is to create 36 ATPs. Those 36 ATPs are produced in one of 2 ways. So the first way is, it's a really long name. Let me write it out first. Now, no one's going to remember that name, so we can just call it SLP for short. And although that's not the official short form, right? But you can use it. Yeah. So this is substrate level phosphorylation. Substrate level phosphorylation. Just going to put a star here. 2-phosphorylate a molecule means to add a PO4 to it. Right? If you add a phosphate group onto a molecule, you are phosphorylating the molecule. For this to happen, we need three things. We need an enzyme. We need a fuel or donor molecule. And this guy has an extra phosphate that it doesn't mind giving it away, donating it. Does anybody remember the empty version of ATP? If you use up the energy in ATP, what molecule does it become? A what's now? ADP, exactly. ADP, right? So here we're gonna have an ADP molecule. Right? This one is ADP, which means it has two phosphates, right? So basically, the ADP is spent. Right? It's empty now. It has no juice in it, no energy. So what happens is the enzyme, I take a look first before you copy, the enzyme will take the phosphate from the donor and put it onto ADP. When you add the P back on ADP, what's the end result? What are you going to get back? ATP, right? Okay, so that's what this process is. Okay, substrate level phosphorylation, okay, is where an enzyme transfers a phosphate from a donor or fuel molecule, doesn't matter what you want to call it, onto ADP to create ATP. Yeah.
I think first is ADP have like the part
ADP?
Yeah.
Two. D means di. Di is two. Like carbon dioxide has two oxygen. Okay, so diphosphate has two. Right, so basically, that's what it is. Now, this is not a popular method, okay? Only about 11% of ATPs are produced this way. 4 out of the 36 are produced this way. Okay? So very little. Nevertheless, it's it's better than nothing. Okay? Oops. Sorry, I have to take this. I will be right back. Okay, so only 11% of the ATP are produced this way. Okay, not a lot, but not nothing either. The more common way of producing ATP is through the second method. So second method is called oxidative phosphorylation. Oxidative phosphorylation. And to explain this is gonna take like half an hour, so we'll do that later. Okay. This this happens at the ETC, okay, which is stage 4. Okay. Majority, okay, roughly like 90% of ATP are produced this way. Okay. Basically, it involves creating ATP using oxidation reaction. Okay. So I'll explain that probably not today. I don't think we have time for that today at all. But that is that. Well, maybe, I don't know. We'll see. So that's the overview. All right. So let's pause for a second and talk about the lab. Okay. So if you just came in, there are some handouts here. And now I put you guys into groups already according to what you signed up for. But it's a good idea to take a look and see if I made any mistake. If you want any changes, feel free to let me know before you go today. Like I said, the lab will be on Monday. It's gonna take probably the whole class, right? You are not gonna believe it, but the most time-consuming part of the lab is boiling the water. Okay, you have to boil the water up to 80 degrees, and yep, it takes very long. But don't worry, like I have a cheating way to do it. Like if if it's really taking too long, I'll come and give you boiling water from a kettle. Okay, you be boiling it from a hot plate, but I got the electric kettle. Right. So now what do you have to do for the lab? Well, for the lab, the procedure is in pink. Okay. Like I said before, you really need to read it through before coming to the lab. You can't just read it on the spot and figure out what to do. Okay? People who did that in the past, again, get very bad data, and it's just not gonna work out. Okay. So read it through. I think it's quite clear how it describes it here in the procedure. There are pictures that shows you what you need to do as well. Okay. If you want a color version, is already on eCentennial. Okay, so please feel free to download it. I would recommend summarizing this, maybe doing a flow chart, and so you're able to efficiently do the work. So when you come in on Monday, I will have the station set up. All the equipment will be at the bench, much like what happens in chemistry class. And it's a good idea to talk with your group members and see who's going to be doing what. Okay. So for example, you need someone to be preparing the water, okay, boiling the water. You need someone to be labeling the test tubes. You need someone else to be cutting the beetroot. Okay. So divide the work so that you can work efficiently. We have digital thermometer now, which is great. We only got this recently because we found out people actually don't know how to read thermometers. The traditional ones. Okay, so this is much easier for you guys now. The digital ones sometimes do run out of battery. So check it as soon as you get there. If that's the case, let me know. I'll replace it for you. Over here, the green sheet is the lab report. Okay. So each group has to hand in one copy of the lab report. This is just a rough copy. This is for you to fill out as you are doing it in the lab. What you have to hand in is like a typed up version of this, and the lab report, the digital one, is also on eCentennial. Okay, so hold on, let me try to find the lab here. Lab report. Do you see it? Oh, there it is. There we go. So you can do the rough copy together and then, work on the good copy also together, right? Because I only need one copy per group. Everybody's going to get the same grade for the report, okay? So some questions you can actually do before coming to the lab, right? Once you read the procedure, you should be able to answer some of these questions. For example, what is the purpose of this experiment? So the purpose of this experiment is to investigate the effects of temperature on beetroot cell membrane permeability. Okay. I give you permission to copy it exactly as it is, okay? So that's 1 mark. You're welcome, right? But it serves a purpose, right? It shows you how it's done. Right? The answer should be appropriate to the amount of marks that's given. Right? If it's only 1 mark, don't write me a novel, okay? Just try to keep it clear, concise, and correct, okay? So that's what we're gonna do. Write a hypothesis for this experiment. A lot of people still, maybe struggle a little bit with the hypothesis. Remember, hypothesis is not a question, okay? Hypothesis is a tentative answer, so it should be a statement. All right. So if the purpose is to figure out the effects of temperature on the membrane permeability, your hypothesis should be a prediction about the effects. Okay. I will give you a sample answer that is not good. Okay. Many people do it this way and then they don't get 4 marks. You're going to tell me why. Okay. Here is the answer they would put. Temperature will affect the membrane permeability. Okay, so they put that for the hypothesis. Higher temperature will affect the membrane permeability. Why might that not get you 4 marks? Or do you guys think that's a great answer? Let me say that one more time, okay? I'll type it out. High temperature. Don't copy this down, it's wrong, okay? High temperature will affect membrane permeability. Why might that be not a good answer? Because it what? Well, I mean, part of it is to predict the result though, right? So it's not so much that, but good try. Saying like how it will affect. That's right. Yeah. So it doesn't really predict how, right? Like, does higher temperature increase the membrane permeability? Does it decrease the membrane permeability? So you need to state that when you write the hypothesis. You guys understand that? Okay. Now, only half of you are listening. So make sure you go home and go through these questions and maybe watch the recording so that you can do well on the quiz, on the lab report. Okay. How much is the lab report worth? Do you guys know? 10%. Okay, that's a lot of marks, a lot of free marks that you can get. Okay, the course is only going to get progressively more difficult. Right? Trust me, second half is quite difficult compared to the first half. And so you want to get as many marks as possible on these things. Okay. So very standard questions, independent, dependent variable, standardized variable, so on and so forth. Okay. Over here, in the data and observation, it says you can write on the sheet, okay? As I'm explaining to you, you can jot down notes, okay? Because you're not handing this in, right? You have to type it up to give it to me, right? Okay. This is just a rough copy, right? Okay. So it asks you to insert the picture you took during the lab here. So at the end, when you have completed your experiment, you need to take one picture of your results, the test tube, basically. Okay, just one. Right? A lot of people document the whole thing and then they end up printing a lot of pictures and putting in the report. Like you're just wasting ink and money, right, to print them. So just one picture, and it's going to be 2 marks. And you want a picture to be clear that I can actually see, the labels and everything. All right. If you don't have a phone that takes a good picture, or no one does in your group, which is unlikely, okay, you can ask me. I'll take it for you, okay? But you need to have a picture. Everything else is pretty self-explanatory. This is graphing. All right, if you don't know how to graph, you can scan the QR code or click on the link. And I'll show you how to graph it using Google Sheets. Of course, you can do it with Excel as well, if you have Excel on your computer. But Google Sheets are free, right? So you want to copy and paste your graph here. It is 4 marks. So a lot of people lose marks on the graph because they forget to label the axes. Okay, you have to make sure to know what goes on the Y and what goes on the X. Okay, so you'll be making two kinds of observation in this lab, qualitative and quantitative. If you're graphing it, which one is it? Is it quala or quanta? Quanta, exactly. Okay. And then there are some conclusion questions. These are the ones where you have to think a little bit about it, okay? I will post some videos after the lab for you to watch, like my videos. And those videos will give you some ideas as to how to answer these questions, okay? Don't Google them, okay? If you Google them, you're gonna most likely end up getting like wrong answers. And that's it. Okay, very straightforward, I hope. But very important to read the lab. Okay, let's take a 5-minute break. We'll resume in 5 minutes.